This video is for The Living Earth, Unit 4.3, Transcription and Translation. Today's essential question is, how is the genetic information transcribed from DNA and processed into functional mRNA? Today, we begin our exploration of one of biology's most fascinating and fundamental topics: how our cells use the genetic information stored in DNA to produce proteins. These proteins don't just build our muscles or make up enzymes, they perform nearly every function our body requires. This process begins with transcription, the first step in a two-part system that turns genetic code into functional proteins. Think of your DNA as a cookbook locked away in the vault of your nucleus. You can't take the whole cookbook out, but you can make a temporary photocopy of a recipe. That copy is RNA. That's what transcription is, the process of copying instructions from DNA into a form that can be used to make proteins. So let's begin by comparing DNA and RNA. While both are types of nucleic acids, which are large molecules made of smaller building blocks called nucleotides, they differ in structure and function. DNA, or deoxyribonucleic acid, is double-stranded and stays in the nucleus. It stores the complete set of instructions for making all the proteins in your body, meaning your genome. RNA, or ribonucleic acid, is single-stranded and is temporary and is a working copy of one gene. It can leave the nucleus and deliver the message to the cytoplasm, where proteins are made. Also note the different pair differences. DNA uses thymine, represented by a capital T, but RNA uses uracil, represented by a capital U. This is important for recognizing RNA sequences, and it helps the cell know it's working with a copy, not the original instructions. Structurally, DNA and RNA differ in three major ways. The sugar in DNA is deoxyribose, while RNA has ribose. The lack of one oxygen in DNA makes it more stable for long-term storage. DNA is double-stranded, with base pairs forming a ladder-like structure, while RNA is single-stranded, which allows it to fold into various shapes for different functions. As mentioned before, DNA uses thymine and RNA uses uracil, which pairs with adenine. These structural differences allow DNA to serve as a stable storage molecule and RNA to function as a flexible messenger and tool in protein synthesis. So let's go step by step through the process of transcription. Inside the nucleus, an enzyme called RNA polymerase recognizes a region of the DNA called a promoter. This promoter is a special sequence that tells the enzyme, hey, start here. RNA polymerase then unwinds a portion of the DNA and uses one of the strands, which is called the template strand, to build a new complementary RNA strand. It does this by adding RNA nucleotides that match the DNA base pairs. A pairs with U, T pairs with A, C pairs with G, and G with C. Once the polymerase reaches a termination sequence, it knows to stop transcribing and then releases the newly formed mRNA molecule. This process produces an RNA strand that is complementary, not identical, to the DNA template strand. RNA polymerase is truly a remarkable molecular machine. It performs four critical functions: recognition, in which it detects promoter sequences to know where transcription should begin. It catalyzes. It forms the bonds between nucleotides, linking them into a growing RNA strand. Proofreading. Although not as accurate as DNA polymerase, it can catch some mistakes to avoid faulty RNA transcripts. And then lastly, termination, which is when it hits the termination signal. It detaches from the DNA and releases the RNA. Understanding this enzyme is key because it drives the entire transcription process. Protein synthesis involves three major types of RNA, each with a distinct role. There's mRNA, which stands for a messenger RNA. This carries genetic instructions from DNA in the nucleus to the ribosomes that are in the cytoplasm. It's the blueprint that makes a specific protein. There's tRNA, which stands for transfer RNA. This brings the correct amino acid to the ribosome and matches it to the mRNA codon using its anticodon. And then there is rRNA, which stands for ribosomal RNA. This makes up the core structure of the ribosomes and helps catalyze the formation of peptide bonds between the various amino acids. Each of these types of RNA has a specialized job, and all three are essential to turning genes into proteins. Now, before the mRNA leaves the nucleus, it undergoes processing to ensure it's stable and functional. First off, there's trimming, in which extra sequences at the ends of the RNA strand are removed by enzymes called exoribonucleases. There's splicing, in which the RNA, where the RNA may contain non-coding sections, right, that do not code for genes, called introns. These are then removed, and the coding sections that are called exons are then joined together. And then there's modification, in which a 5' cap and a poly A tail are added. These modifications protect the RNA from degradation and help it bind to the ribosome. If each of these steps are not completed correctly, the mRNA may not be translated properly or may degrade too quickly. The genetic code is like a language written in three-letter words called codons. Each codon on the mRNA corresponds to a specific amino acid. There are 64 codons, but only 20 amino acids, which means some amino acids are coded by more than one codon. This is called redundancy, and it helps protect against mutations. The codon AUG signals the start of translation and codes for the amino acid methionine. There are also stop codons like UAA and UAG. These two tell the ribosome when the protein is complete. Understanding how codons work is critical for interpreting how genetic instructions are read. Now, it's easy to confuse transcription and translation, so let's compare them. Transcription is the process of copying DNA into RNA. It happens in the nucleus and produces a single strand of mRNA. Translation, on the other hand, is the process of building a protein based on the mRNA instructions. This happens in the cytoplasm at the ribosome. Together, these processes make up gene expression, which is the flow of information from DNA to RNA into protein. This concept of DNA to RNA to protein is called the central dogma of molecular biology. Not all genes are active all the time, however. Cells use transcription factors to control which genes are turned on or off. Initiation factors help RNA polymerase bind to DNA. Activators increase gene expression by helping transcription complexes assemble. Repressors block transcription, either by preventing RNA polymerase from binding or making the DNA harder to access. This regulation allows different cells to specialize, even though they all have the same DNA. Mutations can change how transcription works. So let's break down a few types. There's substitution, in which a single base is changed. It might have no effect, which we would call a silent substitution. It can change one amino acid, which is called a missense, or introduce a stop codon, which is called nonsense. There can be insertion or deletion, in which there can be a shift in the reading frame, which changes every codon after that mutation. These are called frame shift mutations. There's also a splice site mutation, which can interfere with RNA processing, which causes the wrong parts of the gene to be included or excluded. These mutations and others can be harmful, neutral, or even helpful, which can drive evolution and diversity. Scientists can observe transcription in action using several techniques. There's northern blotting, which shows RNA molecules as they are present and their sizes. There's RT-PCR, which converts RNA into DNA and then amplifies it to detect even tiny amounts of gene expression. And then there's RNA sequencing, where there is a reading of all RNA molecules in a cell, giving a full picture of which genes are active. These tools, among others, help us study gene expression in different conditions, tissues, and diseases. Understanding transcription has transformed science and medicine through gene therapy, which corrects genetic disorders by repairing or replacing faulty genes. mRNA vaccines, like those for COVID-19, they use synthetic RNA to teach your immune system how to fight viruses. And in agriculture, modifying transcription can help crops grow better, resist pests, and survive climate change. This knowledge doesn't stay just in the classroom. It leads to real-world solutions that improve lives. At this point, you should be able to generate a full and appropriate response to today's essential question. How is genetic information transcribed from DNA and processed into functional mRNA?